AJ. It's all right. Hi. It is 9 o'clock on Thursday night. Listen to some of the Mills Brothers. Do you know the Mills Brothers? We could spend the entire 30 minutes rather listening to the Mills Brothers. That on some level might even be better. But we won't. We're going to discuss viruses. Right. So what we're going to chat about tonight is quite an interesting study that was done pretty recently. Obviously, everything has been done pretty recently because it's all just new, right? It's all just begun. This pandemic is only, when did it? December, it's January, February, March, April. 4 months, three to 4 months now. So still very early days. So again, we're just going to be chatting tonight, and a friend of mine asked me if I'd heard about the Santa Clara study, and I had sort of heard bits and pieces here and there about it. And so, what is the Santa Clara study? So basically, look, Santa Clara is, I think, the district or county of California. I thought it was, for some strange reason, South Carolina. I mean, I don't know where my head was. But anyway, it's California. And they addressed one of the biggest issues in basically this outbreak, okay? Well, that was their goal, is to set about addressing this really important question. So that Santa Clara study. Now, basically, their theory was that serology is the thing to catch the king. And I think I think William Shakespeare said that, although probably not. I'm sure that serology was not very big in his day. But basically, what it is, this Santa Clara study, let me just go and, we're going to be vacillating myself, and I have put a couple of slides together. So the really big issue is this equation here, all right? There's a fatality rate, okay, which is trying to work out how many people that are infected are actually dying from the disease. And we know that kind of varies all over the world, and that's probably typical of most viruses. But the way you work it out is you've got to work out the number of confirmed cases, and you've got to multiply it by some factor, which basically tries to take into account all of the asymptomatic people who've been infected with the virus, but are essentially not presenting any symptoms of the virus, all right? So it's a massive question, because there's more and more information coming out suggesting that, we're getting a lot of people that are getting sick from the virus, but actually there seem to be a lot of asymptomatic carriers. Now, how do you work out those people? Because they look well, they look fine, and the problem is the best thing you can always do, is basically what is required is a random sampling of as big a segment of a population as you can possibly do. Now, because then you can see in amongst those who is sick and who is carrying the virus but is not presenting any symptoms. Now, the big issue is that if you try and do that with a nucleic acid test, all right? Now, remember the nucleic acid test. That's essentially what the nucleic acid test is. I'll just quickly go to my blackboard. Is just over here, right? So that's the nucleic acid over here. I don't really need to actually be drawing it, but essentially what you're doing is it's essentially a way of capturing active viruses in the system at the time, all right? But what it unfortunately misses is if you've cleared the virus, you should no longer be carrying the nucleic acid, all right? So there should be no longer, because it's really evidence of the virus. It's a way of detecting the actual virus. And you do that by detecting very specifically its genetic material. Now, fortunately, there's a way around this, a way of knowing at any one time in the population who is currently infected and perhaps not sick, and also who has been infected in the past. And the way that is done is very simply through what is called a serology test. And I think, I know that a couple of weeks ago I did speak briefly about what the serology test is. But essentially what it is in any person who's been infected with the virus, there will be, even if the virus has been cleared, there will be a memory of that infection in the body in the form of circulating antibodies. All right, now we spoke about antibodies as well. So they'll be circulating around, and we can detect those, all right, using what is called a serology test. So that is the idea, is to basically detect these antibodies that are essentially memory antibodies. They remember that there's been a virus infection, and we can fairly easily detect those, all right? And that'll give us, if we do a sampling of the entire population, an estimate of that unknown factor. And that's what we really, really want, okay? We really want that estimate. So that is what this group did. And really, it was actually, first of all, let's put it in some sort of context. It was a group from Stanford University, a large collection of researchers. I'll show you the preprint right now. So let's go and have a look at the quick preprint. It does require me, there we go, okay. Just so you can see what it looks like. It's a preprint, COVID-19 antibody seroprevalence in Santa Clara County, California. That is the name of the title. Those are all the scientists working on it. I'm not going to list them all. They've appeared in the media quite a bit. Jay Bhattacharya has been reasonably prominent in the media discussing the results because the results are huge. I mean, they're really quite fascinating, interesting, potentially very important. It's always important to note that this study is also in the form of a preprint, so it hasn't been published yet. The data has been made available. So what did they find? What they did was essentially they went into the community in the Santa Clara County, where I think there's in the region of 1,200 to 1,400 cases. Am I getting that wrong? Am I getting it wrong? Just let me just check that. I wrote it down here because it sounds low. Hang on, I think it's something we should actually find. So just bear with me, yeah? Results. Yeah, it was in the area of about over 1,000 cases, okay? I can't remember the exact number, but essentially, I'm pretty sure that's right. I should just confirm that. Introduction. So yeah, bear with me, yeah? Yeah, 1,094 cases, all right? So it's not an insanely high number, but it's a reasonably high number. So they wanted to know, over the whole population, what is the seroprevalence? How many of these people actually are infected without us knowing it? Okay, it's a fantastic question and an important question. So they called over 3,000 people, and they essentially subjected them to a serology test, okay? They basically took blood. The way they did this is quite interesting and quite novel, is they put a Facebook ad out into the local county and said, right, come to these various locations if you're happy to participate in the study, and we're going to take a little bit of your pinprick, and we're going to basically take a little bit of that blood and we're going to stick it in this little cassette that we've from this kit that we've purchased from this company who are called Premier Biotech. So it's an American company who supply this product, and essentially people arrive at these various locations in their cars. It's quite important. So people with cars could go, and people who have access to Facebook couldn't go, all right? Then off they go, and they eventually, at the end of the day, capture something in the order of 3,300 valid samples that they then go off and test for the presence of antibodies against COV2 antigens. All right? It's all good, and it makes a tremendous amount of sense. Cannot fault, this is a group that has been doing this kind of work. I did a little bit of looking to see the kind of work they do, and they are public health people. They're working in the area of interests in global health concerns and epidemics. So they are possibly the right people to be doing this. All right. Now there's a couple of question marks, and those will appear a little bit later. And please keep in mind, this has not yet been peer-reviewed. So let's go back to what did they find? Okay, this is what they found. They basically, if you took at the results from all 1.5% of those 3,300 samples were positive, all right? In other words, came up positive for the serology test. They had to do some statistical adjusting, which brought it up to about 2.81%. Now, the really important number down here is that suggests that there's 50 to 85-fold more infections than the number of reported cases. 50 to 85, almost 100 times more people than who are known to be infected appear, according to these results, to be carrying evidence that they've had the virus or carry the virus. They either are not symptomatic or they've cleared the virus and were never symptomatic, but it's a very significant finding. All right, and unsurprisingly, really, really significant in terms of what it could mean. All right. So the key thing here, there are two really important things we need to look at, because these are obviously very significant findings. Now, please, obviously, as we go, if there's something you'd like to ask, feel free to do so. But I think there's two things about this study which we need to look at, all right? Because first of all, the one thing about a study like this, and I, having worked in a laboratory, there are two key things. There's the sampling, how they went about actually getting the samples that they got, okay? Because this is what the actual data look like, really, all right? So this is here's the story, all right? I did this drawing of what the story. There's the whole population, right? Let's get my get a pointy thing. There's the whole population, right? I've drawn it as a circle. These are of that massive population, which is probably 80 to 100,000 people, okay? In the whole population I'm talking about now. Remember, they are not sampling the whole population. They are sampling a fraction of that population. And they're doing it by calling people on Facebook to come to these sites. They can measure their sample, their antibody levels, right? Over there are the actual number of infected people, very tiny proportion of that entire population. And then this is the part that they're trying to measure. Who are the people out there who are carrying the virus or having had evidence of having carried the virus? Who are these people? Can we find them? All right. The way you do that in a statistical study, and let me just be very clear, I want my face to be in the picture for this, am not a statistician, all right? I have a very passing understanding of statistics, but I've encountered it in my research days, the perils of statistics, all right? And I think we all know that they're a little bit perilous, even back to Mark Twain, who said, lies, damn lies, and statistics, all right? I'm not suggesting that all statistics are somehow evil, little monsters going around trying to subvert data. But what I am suggesting, and what is coming out of some of the analysis of this study, is that the design of the study designs are incredibly important. And we've got to remember, in the age of COVID, that who the hell's got time to do a proper study design? We're trying to get some numbers here. Damn it. Okay, so there's a few things to factor in here, all right? Now, this is how you sample a population, right? Very crudely, I'm trying to show what you want to do. Because you can't count the whole population, all right? What you're trying to do is those red dots there, is you want to just sample a representative, as close as possible to a representative of the entire population as you possibly can. Now, that was very challenging in the study because, first of all, it was, I won't say it was done quite quickly by this novel, very clever approach of Facebook call. And as a result, there was quite a significant bias in I. If you look at the whole population, not all zip codes, postal codes were equally represented. So not all races were equally represented. Children, is surprising, were quite well represented because what happened was children would go in the cars of their parents and go to the sampling sites and get sampled themselves, which actually is an interesting confounder, because that you'd expect if someone is carrying an antibody in the household with the child will also carry the antibody. So there are confounders, and they were aware of these confounders. Every time you have to factor in a confounder into your equations, your statistical equations, you end up with the error, the margin of uncertainty, let's call it, creeps up a little bit, okay? Which is acceptable. That's the nature of the beast of statistics. This is really what the sampling kind of looks like, all right? So I've shown this really idealized situation where you get this nice even coverage of the entire population. It'd be wonderful if that happened. It probably almost never happens in statistics, but you try and get it as close as you possibly can. What's more likely going to happen is some zip codes, which I very crudely try to illustrate here, are overrepresented. Some racial groups are more overrepresented because maybe poor versus wealthy, have access to cars, are active on their Facebook. It really is a sampling bias that has the potential to confound the data. Now, they did attempt, and this is important, they did attempt to adjust for these confounders, but it have the potential to skew the data. Even quite small fluctuations can skew the data. So it's important of that, okay? I'm going to just take you back to, I've got this image here. Because being a laboratory scientist, well, I was in my day, I'm not sure you can really see this very well, but now I want to get across to you the sort of enthusiasm and the feeling of doing these kinds of studies, or basically sampling a whole lot of, a massive number of people, 3,000 people in this case. If you count all those grids, actually there's about 3,000. I've colored in about 50 of those grids, because that's actually what they got. They got 50 colored in blocks over there of all 3,000. Now, me in the lab, I would say, this is fantastic. Look what I'm getting here. I'm getting all these fantastic positives. This must mean something. I want to go now and tell the world that we're getting more positives than we would expect to get. So the prevalence is suggesting there's more people who are carrying the antibody than who are not carrying the antibody, i.e. there's more people carrying the virus. A good study leader, and if this goes anywhere near the journals, or I attempt to publish this, I will very quickly get swatted down by a statistician looking at the study saying, your study design, even though these numbers look compelling, actually on a statistical level, they don't necessarily mean as much as you hope they mean, right? So it's really important. The point I'm trying to get across is, again, I repeat, I'm not a statistician, but it's really that study design is really, really critical. And there's another confounding thing. Basically, the other confounder was, and to me, this is a really significant thing. So we have these confounders. The statisticians who looked at it were quite concerned by some of the data they were seeing. Maybe I should just quickly talk about what their concerns were. So I mentioned that like some statisticians, because now remember, this wasn't peer-reviewed, but a really interesting thing happened in response to this preprint. There was almost like a peer review happening off the cuff by quite serious statisticians happening on Twitter, right? And, there's obviously Twitter's not a reliable peer review, just as much as a preprint is not a reliable source of, necessarily a reliable source of information. But, they make the point that the prevalence changes, like the amount of the result changes with even a slight adjustment for a very important little thing here. Okay, and this is what I'm going to mention now. People who think they might have the coronavirus will be in quite a hurry to go and do this test to see if they've actually got it. All right. So that is called there's a selection bias. So the more likely you are to think you're not feeling that well, you'll go and check your antibodies. All right. They did request that previous illness or how you're feeling was a question they asked in the questionnaire. But interestingly enough, they don't mention anything about it in the actual study, which I thought was a bit surprising. All right. I love this little Twitter comment here. From this Carl Zimmer's lab, and he's quite a well-known statistician. Loud sobbing reported from under Reverend Bay's gravestone. Seriously, I might use this as an example in my class to show how not to do statistics. Note that the CI, which is the confidence interval, so the confidence interval specificity includes false positives rates that are larger. So in other words, the error margins in some cases are actually bigger than the result, which is a concern. All right. Having in my days in the lab, if that happened, if I saw an error bar that was larger than my actual data, I would be concerned. I would be a bit reluctant to believe my own results. Okay, so that is important to keep in mind. So it doesn't. I think what's important is it doesn't discredit the study, and I think these guys were aware of some of these limitations. This is part of the rush of doing science in this early stage, but I do think study design may not have got the attention it deserves from what I'm picking up from the comments. Now, me as a molecular biologist, my bigger concern, only because that's where my understanding is stronger, is around the test they used, right? So the test was interesting. They got a test from this company, the actual serology test, and the test they used was very simple. As I previously mentioned, they take a little bit of blood, they put it on this maybe I should draw it, just to give you the principle of how it actually works. So the test really is quite simple. They take blood, all right, serum, let's say, the serum fraction of blood, and they put it onto this strip here, right? And basically they've got antigen here. Antigen is COV2, another fancy word for COV2 protein, but it's COV2 protein that is recognized by antibodies derived from serum, okay, known to react to antibodies derived from serum. And basically what they do is they put serum on there and they test to see if it sticks, all right? So that is the nature of the serology test. If it sticks, then they've got a positive result, all right? Now, obviously, if you're going to develop and do a test like this, you've got to know before you do any of this that the samples that the antigen you're using is a good test, all right? It must be a good test. Now, the antigen they used came from a laboratory or a company in Hangzhou in China. I couldn't for the life of me find too much information about them. The thing that bothered me the most is I actually couldn't find information about what the antigen was they used, all right? And it's really important to know what antigen they used. It's also important to know if they had controls like other coronavirus antigens on it, because other coronaviruses may, and they probably don't, as a lot of studies suggest, but they may react to the sera from a COV2 patient. So these other coronaviruses would be coronaviruses that cause cold, and they circulate in our population all the time. They are there. So it's surprising that they didn't mention the presence of, if there even was that kind of antigen. So there was an omission there, which really concerned me. The other thing is you want to know that when you take the serum from a patient, if it binds and you get a positive signal, what does that mean? So they have to test it. So what they did is, as part of the manufacturer's kit that was developed in this Chinese biotech company, they have in the order of 100, a decent number, like about 150 to 170 so-called positive samples. This would be serum derived from people who have been known to have a COV2 infection. They included those in the kit as a control, so you can test it. So they did those tests. They showed that most of the time, in fact, I think it was all the time, if you got a positive reaction against those test cases, which is great. They also showed that if you take serum that comes from a person who has never been exposed to COVID, and they don't explain how they got the serum. They don't say this in the study, except to say that it's free of COVID, which I have to assume that I don't know, I can't assume anything really. But the important thing is those most of the time did come up negative, not all of the time. So you do those studies so you can calculate a kind of factor which shows you how reliable your data is. Now, they clearly were a bit nervous themselves about using this kit, so they got their own samples from their own freezers in Stanford of cases that they'd already recovered and shown to be COV-2 positive. And they showed that most of the time, again, those samples came up positive when they should have. They also took, and this is a much better sample, or this is the way you should do it, is they had samples in the serum in their freezers, which were obtained prior to the whole outbreak, okay? So they should be definitely free of COV-2, and those came up negative in most of the cases. Now, it's okay to get some false positives and false negatives, because you factor those in, but according to the statistical stuff, the number of false positives they did have and false negatives, they didn't really have enough material to sample to outweigh the impact of having data that was coming up positive or negative where it shouldn't have. So if their sample size was much, much bigger, it would have compensated for the, even though it was a low rate, but it would have compensated for the low rate of positive and negative results. So they didn't really have that, and that is concern. So my real issue with this study is, I think it was an important study, and I think, look, they need to do a lot more studies like it, but they do need to have a much greater certainty around whether these serology tests are actually well defined, all right? Now, it's important to note that this actual serology test they used was actually, banned, all right? Was banned by the FDA. They decided it wasn't, or they hadn't evaluated it properly there. This group had done the study before this ruling had come out. They shouldn't necessarily have used it. That doesn't negate its value, because, and I'll tell you this much, which is quite interesting, quite intriguing. The Australian government, the Australian Register of Therapeutic Goods, this is where I live, they actually have it, this serology test, as the first test, and they have endorsed it. So, it's possible that I just don't have access to the right information, but it's curious that the FDA, the Food and Drug Administration of the States, are not happy with it. And it is curious that the Australian government is happy with it. So I don't know what is going on there, other than we're in early days. I don't know if it's been validated in Australia. I did manage to track down a desperate document, which is the product insert, okay? And this is what comes when you buy the kit, all right? Now, nowhere, even in this vast amount of text over here, does it say what the antigen looks like. And that's really a problem. We don't know what antigen they use. Now, typically, what a serology test would most likely use is antigens to the spike protein. Do you remember that spike protein? That's the one that sticks right out there. And there's our virus. The spike protein being that protein that projects right out of the virus coat and is recognized by a lot of antibodies, right? I've drawn a panel of antibodies here. Right, so both AB1, and these are arbitrary, I've just named them for the sake of it. AB1, antibody 1 and antibody 2, they both recognize different parts of the spike protein, all right? That's quite important, because the spike protein is a large protein. Different antibodies might recognize different parts of it and have different effects. I've arbitrarily said that this antibody here is able to actually neutralize the virus. So that antibody can stop the virus from entering cells. That's not important for the serology test, but it could be really useful later when they're trying to develop antibodies to stop the virus from getting into cells, a potential therapy, all right? There are other proteins that they could use that also have antibodies that get developed against them. Like, for example, the nucleocapsid protein. That's another protein that sits inside, a little bit inside, which is not normally accessible to antibodies, but it might be a little bit accessible because they do seem to get some antibodies towards it. Both of these proteins, the spike protein or the nucleocapsid, might be pretty useful in a serology test. Now, they don't say which ones they use. They are definitely developing a lot of serology tests using the spike protein, using the nucleocapsid protein. So it is being done, and it's curious that they don't mention which ones they are actually using here. I'll just show you an example, just for your own, just to show you that I'm not having you on. Let's see, what is the study over here? That's not that one, not that one. What is that? Bear with me, please. Yes, I think this was one of them. Here we go. Here's a good example. This is a kit they could have used from a company. Again, unfortunately, I can't remember the name of the company. Again, it's interesting how many times companies are developing these kits. But development of a clinical application of a rapid IgM, that's the antibody. IG is another name for antibody, combined antibody test for SARS-CoV-2 infection diagnosis. This one has actually recently been endorsed by, I think it has been endorsed by the FDA. There are actually NGOs out there, a friend of mine today told me, who's working in the industry, that are very carefully looking at all of these different companies and evaluating which of these serology tests are going to be useful, because they have to standardize these things. It's no good that one study's using one lot of serology data, another's using a whole lot of other approaches to the serology. They've got to standardize it, otherwise it gets really confusing. So these are some things we've got to keep in mind. Now, let's see. Yes, and the other thing I just wanted to mention, because in my days teaching virology, I was always loved this idea that, again, I'm going to just show you. I see there's quite a few questions coming up. I do hope I get to them. But these proteins, while the virus is circulating in the body, while it's floating around, they have a certain conformation. In other words, they fold in a very specific way. So the antibodies, some of the antibodies are actually only recognize the virus the way it's folded. And it only folds like that inside a cell or inside the blood of a human being that it's circulating in. If you take it outside and you try and isolate it and use it in a serology test, you can change the shape of it. And sometimes it's one of the reasons that the protein, once it's been removed from the cell, doesn't work as well because its shape's been changed. And I've tried to illustrate it by do you remember these little kits? When you were a kid, you used to make these things here. You open them and close them and do some tricks. I got my son to show me how to make one. So, for example, let's just see that thing says spike. That's the spike protein, right? You take that thing out of the virus and you change it. You suddenly, basically what you're doing is you are changing its shape. And you no longer have the spike protein as it should be. The whole thing is conformationally changed. It's a very crude way to try and show you what I'm trying to say here. But you get the idea. By changing it and taking it out of the cell, out of the virus, you can potentially change the whole structure, which can have deleterious effects on your serology test. But there's usually so many different antibodies that are binding to your antigen that it doesn't really matter all that much. It's just a curious thing that I find really interesting. So I think overall, this study, I think, is an important study. And although there may be issues around the statistics, I think overall it probably is not wrong. It's probably only partly wrong, okay? But it's probably also quite a lot right, that there may be quite a large overestimation. But I think we have to be careful of the numbers. There was another newspaper report that a friend of mine sent me published in the Daily Mail. Alarm bells ring. It's not that one. I think I put it in my slides. Let's have a look here. Yeah, Daily Mail. A third of participants, that's 33%, right? It's a lot higher than the 2.8% that the Santa Clara group found. A third of the participants test positive for antibodies like COVID-19 after giving their blood samples at the street at random. That seems insanely high and does seem possibly, there was no recourse to a publication that I could find. So headlines like that are troubling because, well, they're just troubling because it suggests a really large number, but without any kind of backing, that smacks of very serious, misinformation, or it just doesn't sound right. So, headlines like that should be avoided at all costs, especially if they're coming with, in that sort of form. So, I'm going to, I'm not discrediting the study. I think there are issues. Like, it would have been good if they had given us more information about the serology test, because I just don't know. It's hard to say at the moment exactly what's in it. So that's a concern that I have. And obviously, study design seems to be a potential issue. So I think it's an important study, a novel way of going about getting data. We are under pressure to get data, so I do think it's a group that's whose work should be taken seriously. But again, it reminds us that this should go through some peer review, so that we can see if design needs to be strengthened, etc., etc., etc. So that's all I wanted to say about it. Now, what I would like to do is just see if I can just quickly go through a couple of the questions. And I don't want to keep you for too long. It's already been almost 40 minutes, so let's just do a couple of questions. Martin's saying that the same group is doing, and thank you, Martin, for actually drawing my attention to this study. That they're going to be repeating this with major baseball teams. That's interesting. Does it get even harder to do serology tests if there are different strains of COVID-19 in the community? Oh, that's a really good question. Does it get even harder to do serology tests if there are different mutant strains of COVID-19 in the community? I don't think so. For the simple reason that there's usually quite a large pool of different antibodies circulating in the blood. And so if there are mutations on these spike proteins, which there very often are, there'll probably be antibodies for more constant regions anyway. So those will probably be picked up. Maybe some of the very variable regions, which are usually the ones involved in neutralization. That's the part of the protein that stops the virus. If the antibody binds that, stops it getting to the cell. Those are usually more variable. So I don't think that's actually going to be a major issue. So good, but it's a fantastic question. What happens? Okay, that's, I think you've asked. Tracy, my dear colleague from the University of the Witwatersrand. I'm going to read your question, Tracy. Hopefully it's not going to get me into trouble. Results of the Santa Clara study are consistent with a lot of other studies coming out recently. The first case I'm struggling to open this question. Let's see more. Sorry, it's not letting me open it. So I think you're saying that it's consistent with a lot of other studies, and I think yes, it probably is consistent with there being a higher rate of infection and of asymptomatic carriers. The issue really was around the extent of it and the study design. So I would agree that I would think that this is not an incorrect result, just one that is the issue is more around again, it's probably just boils down to this fact that it's in a preprint form. So the information is, well, the design could maybe have been better, and the serology is just incomplete, at least from a provision of information. Although I should say they did say they had an appendix to the paper, which I couldn't find. So maybe it's all in there, but I don't know. Trace again, do we have any idea if the antibodies detected by these tests would prevent reinfection by the virus? Really good question. I was just reading a paper now, which did actually, as part of their serology test, evaluate some of the antibodies to see if they could neutralize the virus. And there are pools of antibodies that are circulating that will do that can neutralize the virus. So that's quite encouraging. That was, I think, a really, it's a bold study and one that really needed to be done. Leon has just come in here. Leon Perlman is a virologist colleague of mine, pointing out, and he pointed out a paper to me earlier about, and somebody else also has, is that there's some of the strains, there can be genetic differences in these strains now, which have got phenotypic consequences, okay? So when I say that they can make the virus more virulent, it seems like from the in vitro studies. So the next Virology Studio, that's what we're going to be talking about. So if there are no more questions, I hope that has shed light. If I have to, give a thumbs up or thumbs down to the study, I think it's absolutely in general, a thumbs up. It's always a challenge, when it gets filtered through the media. It tends to then, I guess, politicize it a bit, because it comes issues of the value of the lockdowns, which are obviously very important questions that we need to be asking. But that's why the quality of the data becomes so important. But as I repeat again and again, it's that tension between quality of data and getting data out there. So great to talk, and we'll speak soon. All the best to you.